Muchas gracias por la oportunidad de hablarles un poquito hoy sobre mis trabajos anteriores. Y quiero empezar antes de todo dar la gracia al EBD para apoyar nuestro sabático aquí, y especialmente a Monse Vila y a Pedro Jordano, quienes han sido sumamente amable y atento como hospederos para nosotros. Entonces, muchísimas gracias. Ha sido un placer y un honor también estar relacionado con estos grupos científicos y, claro, con todo el instituto que es famoso por la calidad de su ciencia. Entonces, muchas gracias por eso. Today, I'm going to talk about an aspect of my research program that has to do with interactions of pathogens with plants and plant communities. And all of this work has been done collaboratively with Greg, as Monse told the story of. This is actually how we met. It was starting to work on these questions, and so this is really his talk as well. Pathogens can be important drivers of community structure. They can have big impacts on communities, and we know this from examples in animal systems such as BD in frogs, as well as white nose syndrome in bat communities. Unfortunately, we also have a lot of examples from plant systems, particularly forests. For example, right now in Denmark, in parts of Denmark, over 95% of the individuals of one of the dominant tree species, ash, are being killed off by ash dieback. And where we live in California, in the central coast of California, sudden oak death caused by the oomycete pathogen Phytophthora ramorum is having big impacts on some forests there because of the way it differentially affects different host species. So in disease ecology, we want to understand when pathogens are likely to have these large community-level impacts. And in order to do that, we need to understand what determines variation in disease pressure across species. So today I'm going to be talking about two aspects of work that Greg and I have been doing. The first is looking at using observational approaches to look at how numerical rarity and phylogenetic rarity intersect to influence disease pressure at the community level. And then towards the end, I'm going to put on my invasions hat and talk a little bit about the implications of this work for when you expect to see escape from natural enemies in invasive species. And we'll talk about sort of the conceptual basis for that later. Okay, it's a general assumption, an important fundamental assumption of disease ecology that disease increases with abundance. So greater abundance or density can lead to greater pathogen pressure. And in plants, we have quite a lot of empirical support for this. There's a couple of different mechanisms that can lead to this. First is density-dependent transmission among hosts. And the second is an evolutionary process where natural selection can cause pathogens to infect, to be better at infecting more common hosts. As I said, we have examples of this from many plant systems, particularly managed systems, single species studies in managed systems such as agriculture. What we don't still have a very good sense of is whether these density-dependent effects of disease translate into patterns across species within a community. So is it true that the more dominant or more common species in a community experience higher disease pressure? There's actually a couple of reasons why you might expect not to see a clear pattern like that at the community level. One is that if the numerical dynamics of the host are tightly linked to the pathogen, you might expect that the rare species are the ones that are experiencing the strongest disease pressure. So it could be the other way around. And actually, Greg is going to be talking about the possibility of pathogens forcing species to be rare when he gives his seminar in the winter. The other possibility is if the numerical dynamics of pathogens and plants are closely linked, you'd expect that there might be cycles of abundance in the two. And so any snapshot in time, you may not see a close connection between density and disease. But this pattern is really very poorly studied until now at the community level. So that's what we set out to do. We studied for our studies, we used the grassland community associated with the University of California, Santa Cruz campus. So this is a photo from our campus. As you can see, it's very beautiful, and I hope all of you will come visit us when we go back there next year. We had, across this landscape, 10 randomly placed sites. Each site was sampled with a 20-meter diameter circle. The entire community has about 43 species. And those species range in cover from 0.01% cover all the way up to 26% cover. So 3 orders of magnitude of difference in terms of the abundance, relative abundance of the species. So what we did was in each of the 10 sites, we estimated the relative abundance of all the species, and then we wanted to relate that to the disease pressure on each species. So how did we do that? For each species in every site, we randomly selected between 5 and 20 individuals. And because we didn't want to have artifacts associated with whether we were looking at young leaves or old leaves and the life history of different plants, we took the entire individual, and then we dissected it out into its individual leaves. Then we were able to scan those leaves and use software developed by the American Phytopathological Society to estimate the amount of disease tissue on the leaves. We did not include any damage caused by herbivores in this study. Okay, so what we have now is the x-axis is the relative abundance of the plants, and we're trying to predict the amount of disease, and we found that in fact the more common species did experience higher levels of disease pressure. In other words, there is a rare species advantage in escaping pressure from pathogens in this system. There's a couple of important implications of this pattern. The first has to do with an idea that's been debated a bunch in the literature recently, having to do with how pathogens relate to species diversity in communities. And in particular, there's this idea of the dilution effect, where people have argued that maybe one possible value of biodiversity and a risk of biodiversity loss is that having more species in the system reduces the chances of large disease outbreaks. And as you can imagine, this is related to density-dependent disease transmission. So we did find that more common species had stronger disease pressure, which potentially could suggest that increasing biodiversity of this system could decrease the level of disease pressure. The second implication has to do with the maintenance of diversity. So density-dependent disease is expected to promote species diversity among the hosts. And this is an idea we often call the Jensen-Connell hypothesis, and Matt Leibold mentioned this a couple weeks ago as well. The idea is that as species become more dominant, they are affected disproportionately by their enemies, their pests and pathogens, and that reduces their competitive ability and allows rarer species to persist in the system. Now, the idea of the Jensen-Connell hypothesis and everything having to do with the maintenance of diversity and the role of pathogens assumes that pathogens are host-specific. So it has to be, the pathogens have to be host-specific for this to work. However, we know that most pathogens are not specialized to a single host species. There's no pathogen that can infect all organisms, but at the same time, there's also very few pathogens that can only infect a single species. So that leads to 2 important questions. So in order to understand whether species diversity will reduce disease, that is the dilution effect, and whether disease will promote species diversity, we need to know 2 things about pathogens. The first is how many host species can they infect, that is how specialized their host range is. And then secondly, which host species can they infect, and can we use that to predict how pathogens are likely to affect plant populations or other host populations. Okay, this is a problem that Greg has actually been working on for a number of years, along with our collaborators from USDA, the United States Department of Agriculture, Animal and Plant Health Inspection Service. And what they have worked on or done is collected a huge database with 210 different angiosperm plant species, genera, and all of the pests and pathogens that have ever been recorded on those species. So it's an incredible, rich data set. And what they've been doing is looking at the patterns of how specialized are different kinds of pests on plants, and then how is their structure in which hosts different kinds of pests and pathogens are on. And we're going to talk about this again in a little while, but at first I'm going to use their data to just give you one factoid, which is that only about half of microbes are specialized at the genus level. So this isn't even at the species level. So it's the majority of species of pathogens are actually not perfectly host specialized. Okay, what does that mean? Well, if you imagine a little plant community here with 6 different species in it, the fact that pathogens are not perfect host specialists means that we expect to see a lot of pathogen spillover. That is, pathogens moving from one host onto another. And because of what I talked about earlier with density increasing pathogen communities, or populations, we might expect to see something like this, where more common species are going to be responsible for more of the pathogen spillover, and therefore having more of an impact on other hosts in the community. Okay, so which host species? So we now know that most pathogen host ranges are, we're going to talk about exactly what these look like, but we also want to know which host species are infected by different kinds of pathogens. And in order to address this question, I'm going to start with a little digression. So another thing that we've been thinking about a lot is the phylogenetic signal of traits and interactions of species. And I know a number of people in the room have also been thinking about these same kinds of things. So phylogenetic signal means that closely related species are more likely to share a trait than are distantly related species. In other words, that traits are evolutionarily conserved. And Greg and I, for much of this fall, have been working on a manuscript for the annual review of phytopathology that we just submitted day before yesterday. So thank you to the people in the room who helped us make that paper better. In that paper, we spent a lot of time reviewing the traits of plants and microbes that might influence their interactions. And what we found is summarized here. These are all the different traits, many different kinds of traits, leaf traits, stem traits, root traits, ecological and life history traits of microbes, where people have studied the degree of evolutionary conservatism of the traits. And all of these traits in red are ones where a significant phylogenetic signal has been found. So places where these traits we know are conserved. So what you might expect is that if these traits are phylogenetically conserved, and one way of looking at that is to look at how the trait changes with phylogenetic, or the distance in the trait mean changes with phylogenetic distance between 2 organisms. If all of these traits that potentially influence the interactions between plants and pathogens are conserved, then we might expect that the pathogen host range itself is conserved. So the susceptibility to a particular pathogen might depend on the distance between 2 potential hosts. Okay, so going back again to the study by Greg and colleagues from the USDA, they did this kind of analysis with their data set and found that, in fact, there's strong phylogenetic signal in the probability of sharing many different kinds of pests, both microbial pests as well as herbivores. Okay, so most pathogens are not specialized to a single host, but there is strong phylogenetic signal in host ranges. And so the question is, can we use that information to predict how diseases might affect plant communities and how pathogens may move across hosts and communities? It suggests that in addition to there being a rare advantage, a numerical rare advantage, that we might also see an advantage of being phylogenetically rare in the community. And that's what Greg and I set out to test in our system. Okay, it suggests that community structure matters to disease. And that's because if you now take this plant community and you add its phylogenetic structure on there, you might expect that pathogen spillover would be more likely, or maybe only happen, between host species that are in the community that are closely related to the focal species that you're interested in. And we tested this in our grassland. The first thing we did was we took the 43 plant species and we put them onto a phylogenetic tree. We did this using a super tree that Greg put together that actually has stable age structure so that you could use it for any list of plants for any kind of study. So a wonderful, fantastic tool that is available online, and the R code is right there and everything. So I'm sure you're already thinking about ways that if you poor animal people worked on plants, you'd be thinking about all the ways that you could use this database and this approach. So what we did was we got our community and we built this phylogenetic tree, which allowed us to look at phylogenetic distances among every pair of species. And then we made predictions about how the level of phylogenetic isolation of each individual species would be related to disease pressure on that species. So for each individual species, you can look at a number of different potential metrics of phylogenetic isolation. You could look at how closely related you are to the closest relative. You could look at how closely you're related, your mean relatedness to all of the plants in the community. We have found in our studies that the 10th quartile turns out to be kind of the sweet spot of where you see the most differentiation among plants, species, and what turns out to be most predictive. So if you look at the distribution of phylogenetic distances to all other species and you take the 10th quartile, you find that, so each circle here is, again, a plant species, and they have error bars because we estimated them in the 10 different sites, and you find that percent disease is lowest for the plants that are most phylogenetically distinct from the other plants they grow with. While you're contemplating the meaning of that graph, I'm going to address a concern that some of you might already be thinking. In particular, so we worked in this grassland, and you might think that there could be correlations between species abundances and their phylogenetic relatedness to other species. So if you think about grasses, right? So you're in a grassland, there's a lot of grasses, they're all closely related, they might also all be really abundant. So we had to make sure that this was not just an artifact of species abundance. And just turned out that in our particular data set, phylogenetic distance was not correlated with abundance. So these two things are acting independently in this particular system. Okay, so phylogenetic relatedness or phylogenetic isolation influences disease pressure, and we can actually do an even better job of thinking about how these dynamics might work quantitatively by going back to our model, or quantitative model of host sharing from the database that I mentioned before. So we know from that database that we can do a pretty good job of predicting the probability of sharing a pathogen between two species based on the phylogenetic distance between them. And what we did was we took this curve estimated for fungi, because we know from various previous studies that most of the disease effects in this community are caused by fungal pathogens as opposed to viruses or bacteria. We take that and we can predict the probability that any given host might share a pathogen with other hosts in the community. And that allows us to calculate a community-wide host abundance, meaning that each species in the community, so if we take one focal plant species, and each species, we calculate the probability of sharing a pathogen with that species based on the phylogenetic distance between the two species and the abundance of that species. So we can calculate, pull that all together, and make a quantitative prediction about how much disease pressure that focal species should experience. So we did that, and this is just to show you that we actually got statistically significant predictive power out of looking at the abundances and the phylogenetic distances of all the other species, not even including anything about conspecific density. So we know that that's an important factor. But the best predictor was a model that included information about conspecific density as well as information about community context. So both of those things contributed significantly to the model. Okay, so just to kind of really clarify exactly what we're looking at here, I'm going to go through a couple of heuristic examples. So if you look at the species in yellow, so here I'm showing the abundance, so local relative abundance and phylogenetic isolation and their effects on disease. So the yellow species are these super abundant plants in our community, and they have high levels of disease no matter how isolated they are phylogenetically from everybody else. However, you have these other species like Metacogo and Traxacum campilodes that are really rare in our community, but see pretty different amounts of disease pressure. One of them is very phylogenetically isolated, and the other one has close relatives, and the one with close relatives experiences higher disease pressure. Similarly, we have two species that are kind of intermediate levels of abundance, Schultzia and Nacella, and those two species, again, see really different amounts of disease pressure. The one with high disease pressure has more close relatives. Okay. So numerical and phylogenetic rarity both contribute to disease pressure. And now we're going to talk about how this might be important for plant invasions. So it was actually really fun for me this summer. We went to Denmark, and I got to see Rosa rugosa invading the coastal dunes there. And that was particularly interesting to me because Ammophila arenaria, which is an important native species in this area, was actually a terrible invasive plant where Greg and I have worked. So it's just an illustration of how one of our major questions of invasion ecology is why do certain species become really bad invaders? And a major hypothesis is that some of them leave their natural enemies behind in their native range and therefore are released from pest pressure. And of course, Montse is the person, I think, who did some of the earliest and some of the best work on this question. But it's still kind of debated how important it is. And there's some interesting phylogenetic aspects to that question as well. So this is a paper by J.B. Gillett, goes back to 1962. This is pre-Jensen and Connell, and it's a paper that Greg always wants everybody to know about. And Gillett said, if species or genus A supports a population of pests which can also attack the related taxon B, the latter will find it more difficult to spread into the area occupied by A than into an area not occupied by any closely related taxon, and therefore relatively free from its pests. So he was making the argument early on that release from pests might be related to the ability to spread into new areas. This is related to something called Darwin's naturalization hypothesis, which is that exotic taxa that are less related to native ones might be more likely to become naturalized or invasive. And in fact, Sharon Strauss and colleagues found evidence for this in California grasslands. So we wanted to ask the question of whether introduced species are more likely to escape their enemies when they're phylogenetically rare. And that could be one possible mechanism for Darwin's naturalization hypothesis. So if you are more phylogenetically distinct from a local community, are you likely to be able to become invasive because you don't share as many pathogens with local residents? We tested this in our grassland system with an experiment in which we took 44 novel host species, so plants that had never been in California. So I'm going to zip to the end of the story and say, of course, we took them out. We didn't let them go to seed in the field. Okay? But we wanted to look at how novel hosts would act in the community. So we went to native plant nurseries across the country and selected plants that were not horticultural. So these were not species that had been bred for either pathogen resistance or anything like that. But they were plants that had never been in California. We selected them across the tree. So these red dots are all of our experimental species. We selected them from across the tree of the angiosperms, and we specifically picked a range of amounts of phylogenetic relatedness to local species. So some of these were plants that had close relatives in the community. Other ones were ones that had nothing closely related to them. We put them into randomized arrays. We put them out into the field, and we let them grow for 3 weeks in the field and collect pathogens. We did disease assessments on them in the field. We used our model to predict how much disease each plant species would experience. So again, this is based on a global database that has nothing to do with our local plants. But we were able to predict based on the composition of each of our 10 sites, which we knew in advance, and the relatedness of each of the local plants to each focal novel host. We were able to predict how many pathogens they should share with local plants. So basically, we did this. We calculated the integrated probability of sharing pathogens with all resident species. And this is what we got. So we found that disease pressure on the introduced hosts was strongly predicted by their phylogenetic isolation from the community. And so basically, you have this group of species, so everything that has a lot of close relatives and therefore has high phylogenetically predicted disease had a lot of disease. And all of the plants that had little disease were ones that had very few close relatives. So this has strong implications for the way we understand interactions between local pathogens and potentially invasive plants. But it also has other implications for community structure and community assembly. So this gets me into another area, which I know a bunch of you are also interested in, which has to do with phylogenetic structure of communities. So there's basically two different kinds of approach, or two different kinds of processes and a whole range of things in between that can influence the phylogenetic structure of communities. The first is at the large scale. So if you think of this being an environmental axis, say, of habitat, and this is a community of plants that have this is the regional pool of species and their phylogenetic relatedness. What we expect to have happen is that when you go from a regional pool of potential species to the species that coexist within a particular habitat, only the species that have the right traits to live in that habitat are going to make it. And that's called habitat filtering. That's when you compare the regional pool to the species that you have in a particular habitat, then you'd expect the phylogenetic distances to be shorter than expected from the whole regional pool. So that's the first step. And then the second step is happening at a smaller scale, where in assemblages where you have species interacting directly with each other, you might expect that if competition is stronger among close relatives, which we know it is in some cases, or if competition is stronger when species share traits, which we know it is in some cases, we expect to see overdispersion, that is, species that are less closely related, making it into the same assemblages at the local scale. Okay, so one of the things that's interesting about this, so this is what we call limiting similarity, and we call it that instead of competitive exclusion because competition is one mechanism that can lead to that, but pathogens and other pests can also lead to that through apparent competition. So shared enemies can cause a reduction of coexistence among species. So in that case, the case of limiting similarity, we expect to see longer phylogenetic distances than expected. As you go from the grassland, the number of grassland species possible, to the species coexisting in one single circle, small site. Okay, so how do we measure that? Well, there are a lot of different metrics for phylogenetic diversity of communities. You could look at the mean phylogenetic distance between any two species. You could look at the median. You could look at the nearest the distance between each focal species and its nearest relative. So basically, you could look at it if you think of these are the plants, all the vascular plant species in the Santa Cruz campus. You could look at the phylogenetic distances among all the species, or you could take a quantile approach where instead of just looking at the median or just the closest relative, you look across the whole range. And this is an approach that Greg and I are really like. We think everybody should do it this way because it gives you a sense of the phylogenetic diversity across that whole range, and it allows you to distinguish among differences at different degrees of phylogenetic depth. Okay. So if you take that approach, so here you've got the quantiles of phylogenetic distance, and I mentioned to you before that the 10th quantile turns out to be kind of the sweet spot for our other studies. In this case, the gray areas here are the 95% confidence intervals of simulations where you take, in the first case, the entire regional pool of all of the plant species on the UCSC campus, and then you compare that to so you take random subsets of that to create grassland communities of 43 species that are selected at random. And then you calculate each of the different quantiles of phylogenetic diversity of the community. That allows you to compare it to the actual observed level. And what we find is that as you go from the regional pool to the grassland, you do see shorter distances than expected, which is consistent with habitat filtering. So then you can take your 43 grassland species and you can take random subsets of those that are set to the same number of species that you see in each of the 10 local sites, then compare those local site metrics to your random 95% confidence intervals. And you can see that especially for low quantiles, so for sort of within family level phylogenetic relatedness, we do see evidence for limiting similarity within our local assemblages as well. So as you go from the grassland species to the local sites, you get longer distances than expected from random. Okay, so pathogens seem to contribute or potentially could contribute to phylogenetic diversity of communities. Many different mechanisms could be contributing to this limiting similarity, including plant-plant competition. So I don't want to give the impression that we think that the pathogens are doing all the heavy lifting here. But it is interestingly consistent with what we find about the role of shared pathogens among hosts. Okay, just very briefly want to mention what we're going to be doing this year. We're really excited to be here, and we have so many ideas we want to explore. We actually have, in addition to all the work that I've been talking about on patterns of pathogen damage, we actually collected leaves from all of those plants. All of the host species in the 43 grassland 10 plots, and we also have all leaves from all of those novel hosts as well. We took those leaves and we used Illumina sequencing to figure out what all of the microbes were in those leaves. So potentially, our hope is that we're going to be able to build a plant pathogen network, which has actually not been done very often, to look at how the different pathogens in our system are linking different hosts, and in particular, whether our predictions about the introduced hosts picking up pathogens from their close relatives actually turn out to be true. So we're going to be able to test assumptions about pathogen host range and look at the links between hosts through their pathogens, and it's all really exciting. So in conclusion, relative disease pressure in communities increases with host abundance with implications for diversity. phylogenetic structure of the community contributes to pathogen pressure on individual species, and that has implications for phylogenetic diversity of communities and the maintenance of broader levels of diversity. And finally, pathogen spillover influences which introduced species will escape their natural enemies. And with that, I think I'd say thank you very much for your attention.